would like to mention that this is organized by Am I sharing my screen?
No, I'm sharing it. I'm not sharing it.
Sorry.
No.
Welcome again. Thank you for attending. So I would like to mention that this workshop is organized by LifeWatch, which is the infrastructure that continuously generates biodiversity and ecosystem data through local observatories and creates systems for data integration. I'm at the Flanders Marine Institute, which is the coordination and information platform for marine and coastal research in Flanders. As part of the Empowering Biodiversity Research Conference, there are many more workshops. If you go to the slides, you can check them more on how to register. There are many different interesting topics. Fair data, GBIF, and how to do data management in research. So today we know that we have several sources of information, right? And the question is, where do I go? Because there are so many. And how can I obtain a flow of data that allow me to work in the most efficient way possible? This by accessing data that is already standardized and that is in the same format and obtained from the same source will help us to go through it in an easier way and in a faster way. And there are many users of this data, of course. There are users from academia, industry, funding agencies, but one that we are going to be focused today is the scientific community. And when making scientific questions, the scientists ask themselves questions such as, do I have enough data? Where can I find more? From which region of the world? And how can I make sure that I have the same species as my colleagues? And how can I link this data to environmental data? So in this workshop, we are going to learn how to answer these questions. We are going to learn how to access and obtain standardized data held in different European initiatives. And we are going to be working with representative species such as the codfish and the anguilla. Hello, my name is Laura Marquez. I am a science officer at Vliz, and I am the helper of this workshop organized together with Salva Fernandez.
Hi, I'm Salva Fernandez. I work for the Flanders Marine Institute, same together with Laura, Lotte, and some other colleagues here. We have been organizing this workshop as part of the Empowering Biodiversity Research conferences, and some of us were last week there. And yeah, so very quickly, I have been 3 years working at Vliz as a data manager. We have been writing our packages to access different databases, and we want to show them here and show you how you can combine them. We also got a lot of help from Lotte Paul, that she's over there.
Hi. Can you hear me?
Yeah, I'm not sure if I can hear you. Yeah, you can.
Then I can just Okay, so hi, my name is Lotte. Okay, perfect. No. Hi, my name is Lotte. I study the IMBRC Masters in Marine Biological Resources, and I'm currently doing my so-called professional practice with Vliz. And in this part, I've been working a lot together with Salva and Laura and helped them in creating this workshop. Hi.
Okay, thank you. Hope you can hear us still. It should be working. All right. You can continue, Laura.
Yes.
Okay.
Okay, so in this workshop, we are going to learn how to standardize your data, how to retrieve more data, and to link that to environmental data. This using the R packages from different European initiatives that are authored and maintained at Vliz by our colleagues. And this is going to be organized in 4 different exercises. So we are going to have 2 breaks in between. And we are going to be carrying on with it.
Yeah, oh, that's it. Yeah. More. Okay. Then before we start with the exercises, I will mention some utilities. Well, first, this meeting is being recorded, so if you don't want to appear on a screen, don't show your camera, just so you know, okay? Because we want to share this this video later with everyone online, so people that couldn't join. Some rules. So this is a collaborative workshop, so we actually copying the style of the INVO coding club sessions that are very interesting. So what we wanna, if you can, well, first open these slides that are in the repository that I sent you already. They are online, so you can just open and go through them. They are very nice to have them at hand. Also, if you can click on this link, this Google document, it's just a place where I would like you to share your solutions to the exercises. Once you are done, you can come here. Then you just please fill here your name. When you're done with an exercise, just put an X or a no or whatever you prefer, and then copy here your solution to the exercise. Okay. So this way, other people can see how you did it, and also we can see different solutions for the same exercise, and we all learn more. Okay. This is especially for the people online. For the people here in the room, you can also just talk to each other. That's great. But of course, you can also share it online. All right. So once again, go to the to the repository that I sent you in the email, and open these slides, and you will find all these links there. All right. More things. To ask questions during the workshop, you can use the Q&A option of Zoom or the chat. We will keep an eye open. If you are here in the room, just say to me, but or to anyone here around. That should be all right. More things. I know some of you have troubles setting up your environment. If you succeed and it's working for you, please just use your laptop. It should work everything fine. All these problems with RM is just to make sure that we are using the same packages and the same versions of the packages, so things are less probable to break. It's very important because many of these packages are in development, so they have a lot of troubles often. But if you had any problem and you couldn't do it, then you can click on this link here, or if you go to let me see. Okay, if you go to the repository and you click here on Launch Binder, it will open. It will take some time, but it will open an RStudio session online on your browser with everything installed already. Okay. So if you had any problems during the installation of the packages, you can try with this. I don't know how many people are allowing this. This is a free service from the Sheffield University, I think. So hopefully there's a space for everyone. But if it is working already in your computer, and in mine is working, you don't need to use it, okay? But if you have any troubles, feel free to open it. Couple more things. Yeah, another thing that I like to use, please always don't just change your working directory. Please use the RStudio files, R projects. Okay, so just click on the RStudio file in the repository. You will find it here. Just this file. If you open it, will open with your RStudio directly, and then we make sure that we are in the same working directory. All right. Have to change this stuff. Okay. So any questions up until now? Anything to mention? Right. Guys, if you can keep an eye open on the chat, it will be great because I don't see anything. All right. Okay, everything's fine. Let's go with the exercises. So in the first exercise, and here is when I mentioned that it's nice if you bring your own data. If you have any data or you want to share it, that's okay. It's just up to you to make it more interesting for your own use case. We are going to use an example data set that should have the minimum information should have is a taxon name, a location, so latitude and longitude, and a time, a date. Okay, that's the minimum information to consider a biological occurrence. That's what, for example, GBIF is normally using, what EurOBIS is using, what OBIS is using. Okay, we're going to use this dataset that is not standardized. In our case, it's going to be a dataset coming from LifeWatch. And we are going to standardize with worms and marine regions, so both for the taxonomical part and for the geographical part. I will explain a bit more now, okay? But just a bit first, I really like this comic thing here. So what do I understand for standards here? It's just, let's try to use the same thing that other people are doing. So some people are thought already on how to classify biological entities, and some people thought already of how to standardize regions of the world. So please, let's try to use these standards that they are already creating instead of creating new things that will bring more problems, okay? But I will explain how we'll do it. How we'll do this. I will go through the three parts of this exercise. That's gonna be like getting data from LifeWatch and standardize with worlds and marine regions. The data from LifeWatch, this is just an example, and this is what I said that you can use your own example data if you want. These are some data that are gonna come from the Belgian LifeWatch Observatory. Actually, I have some experts here in the room, so I'm very afraid what I'm gonna say, but I think it's fine. So these are data that are normally connected either monthly through monthly campaigns that we go on the sea, we collect samples, we come back and we analyze them, or also by tracking data, either with acoustics or other types of tags, okay? This data can be accessed in R with this R package that we have been developing with the Explorer. And for example, it looks like this. You load the library, you wanna get some zooplankton data, so you pass here just a couple dates, and here you get a bunch of data. This is specific of the project, so that's why the column names not necessarily have to be like super explanatory of what they have, but in this case it's pretty obvious. No, we have here a longitude, a latitude, a taxon, and a date, and this is what we are especially looking for in this exercise. It's gonna keep it simple. Of course, you can go far and do more things, but for now, this is what we're gonna do. We get this data on R, and then something that we have to do during this exercise is gonna be important to transfer a normal data frame from R into a simple feature object. What is a simple feature object? It's like a data set, but it has a geometry associated to it. So it allows you, for example, to plot online. So you do here a couple of things, and with the same data frame we had before, now we are able to plot it in a map online that you can see on the web. And even you click on the samples and you get more information out of it. Okay, this is important because we need to do some geospatial operations on the data itself. So to do it, you will see more information later, but otherwise you have here the slides. We use this function from the SF package. You pass your data frame, you assign here what are the longitude and latitude columns. You pass here also a coordinate reference system. I'm not gonna go in detail, but this is just a way of telling you exactly where you are in the world. You have more information here if you need it. That's it. And if you put this remove files, it means that it will not delete to keep them. This example I put is the zooplankton. It's not what we're gonna do in the workshop because otherwise I told you already how to do it. We are gonna do with another data type because there are several data types in this package with the Explorer. That is the Git ETN data. ETN is the European Tracking Network, and this is remote data. Normally it's from fish. In this case, we're gonna get fish, but it could be other specimens. And normally what it's done here, and we also got some experts in the room. They put some acoustic tags, I think. So like there is a tag in the fish itself that emits some sound, and then this is received by some receivers that are put on the sea floor in the Belgian part of the North Sea. And also this is shared with other institutions around Europe. And all this data is shared through the European Tracking Network. There are different ways of accessing this data, and this is one of them through the EurOBIS Explorer. This is all the data that is after the moratorium, and it's free for everyone to use, okay? Now, once we have this data, we have to do a match with WORMS. WORMS is the World Register of Marine Species. It's a database of taxonomic names, okay? Especially for the sea. You can go to the website, you can look it up, you can search for things. And what is important here is that it gives you an identifier for each species name, not for each species itself, because you know that names of the species can change. And this is a very nice thing because all these things are linked and they are reviewed by a committee of experts on taxonomy, and it's always up to date. In R, we can use this package, WORMS with 2 R's, which is developed by the R Open Science community. Use this function here. And you pass, for example, a name that will pass the scientific name of the sea turtle. And what you get here is you get here a bunch of data. And of course, you get the link to the website. You get if the name is accepted. If it's not accepted, you will see here that it's unaccepted, and then you will get actually the valid name here. You get the full classification, Animalia, Chordata, blah, blah, blah. Some extra info. But what is more interesting, the thing that is key for this workshop is this number here. This is the FID, and this now is an identifier of this species name, not the species, but just the name on the website, okay? Through this number, this is very interesting, we will use it later to query all the databases and get more data, to load more data from those using these numbers, okay? So you first can pass whatever number here, Worms will recognize what is the number and will pass you more information back, including this number here. For example, if you open this link on your browser, note how this is going to give you all the information of the sea turtle, but look how this number is the number that identifies to this specimen. Okay. After that, we will have done the standardization with worms in the taxonomical part, but we still are missing a standardization with the geographical part. For this, we're going to use another system of lists, that is the marine regions. Marine regions has different parts, but one of them is the gazetteer. Gazetteer is a dictionary of place names, place in the world, in whatever. Like Belgium could be a name and could be in a gazetteer. Brussels could be a name and could be in a gazetteer. Okay. What is important here, we have this gazetteer for marine names, places in the sea, like seamounts or exclusive economic zones of the world. And we also have the location. Where is that? Where are those places exactly? This information can be accessed in different ways. We are developing, especially Lotte Paul here, she has been developing an R package to access this data in R. And for example, using this function, you can just pass whatever text you want here. For example, we pass here Belgium, and you get all the records in the gazetteer just by giving a name. It will make some match, and here, for example, you will see all the records that contain the word Belgium, okay? If you can see here, you actually have here the names. And in here, you have, again, another number. This number is another identifier unique for this place type and this place name, okay? So it's identifying this place exactly. If you look, for example, at the territorial sea of Belgium, we could get this number, we copy it, and we pass it to the next function. The next function, we just pass this number here. We get the record, and this record is already, as I mentioned, a simple featured object, the same thing, and it does allow you to put it directly in a map, as you see. Here, this is not matching exactly the coastline, but this is a problem of this type of presentation. It should be right in your R session, okay? So you can see here, for example, already in a map, where is the territorial sea of Belgium. If you click on it, you get a bunch of information again. Well, that's it. With this, well, remember that the number is again the same, identifies a specific feature, and this number we will use it later for the databases, okay? What we're gonna need to do in this exercise is we're gonna need to do a spatial intersection just to get records that are within the, in this case, for example, in this example, I'm showing how to do it for the territorial part of the North, territorial sea of Belgium. And simply what we're gonna do is just get the points that are there, okay? So you have some points outside, you have some point inside, we just want the points inside the territorial sea. In R, this is simply do with, again, with the simple feature R package. You do ST intersection, and if the two objects that we did before, the zooplankton from LifeWatch, and now the territorial sea is coming from the marine regions, you do ST intersection, and you get just a few records. No, you don't get all of them. Voila, this is it. This is all the info I wanted to give you, and now we're gonna do the exercises, okay? We are perfectly on time, and we got about 20 minutes to do this exercise, so we will come back at 14:10, okay? If you are in our time zone, if not, in 20 minutes, all right? Not much to, I think all the exercises are very self-explanatory. I will ask you to get some data from ETN using the Widget Explorer package, and then I explain here the rest. If you have questions, write in the chat. If you are here, just shout. It should be all right, okay? The exercises are in this path, all right, on the repository that I asked you to download.
You can show how to access them into the folder.
Yeah, sure. Let me see because I need to. Yeah. But you can start already, okay, while I show this. I need to open yours. No. Okay, if you need some help to open it, if you are already on the on your art studio session and you're already on the repository that I asked you to download, you just need to go to files and go to the path where this is, and then it is in source. Okay, you can also do it from your, if you are in Windows or from the file explorers. Just go to source exercises and you will have them here. Okay, so the first one, 01, standardized. You open this one. If you you look at it, you will see the solutions are here, but I will refrain you from using it. Okay, remember to put your solutions on the Google Doc that I mentioned at the beginning. Hello everyone. So it's time. I'm sorry, I have to press you guys, but we have a schedule to meet. We cannot be in this room forever. And but anyways, even if you are not done yet with the exercise, we will go through it together, and hopefully you can follow along. Also, you have the solutions in the repository, so you can just copy them. All right. Okay. I'll share my screen. My studio session. Okay. So to make things easier, I will go directly to the screens with solutions that I wrote. But of course, these are my own solutions. If you came up with something different, for example, I saw in one of the comments that someone is using the tidyverse instead of the base ARF to do left joins. That's completely fine. Actually, we'll use the plier in the next exercise, but I just didn't want to mention it here to not make it even heavier. So when I do, so one thing first, I like to put my scripts on the left and the console on the right. This is a personal preference. Don't get very distracted. It's just how I like it, so I can see more things in my screen. But most of the people, you have your console down here. All right. Let's just call the libraries that we want to use. And to get the data, one thing that I have seen a problem is that yeah, we didn't mention exactly what's the action what we needed. Normally it's time beams, okay? So just puts all these detections all together in a time beam, like for example 1 week, okay? Next was our projects, we want all of them, and data was asking for a year specifically, 2015. So the best is just to give the whole year here in the 2 dates, okay? If we call this, it will take some time. And voila, we get the data. We got around 11,000 rows. If you go here, you will see a bunch of information. Actually, they are all explained in the package, but you have to look for it. It's on the website of the package, okay? And but as I was mentioning, we have here species names, we have a time, we have longitude and latitude. So we have an example here. If you have your own data set, you can also use it for this exercise, okay? So in the next exercise, I was asking to get first a list of the species in this ETN data. Okay, so if you know typical R, you just write your ETN, you call the scientific name, but of course because it's here is going to be repeated many of them. So if you use base R, you can just do unique. But of course, if you are using the plier, you can use the plier distinct, whatever you prefer. Okay. If we do this and we put it on a variable here, so you can see here that we have around 15 species. Species list and call it. Actually, some of them are not a species like sync tag, but this is what's going to happen with the data very often. Like people put together things that are species, things that are not a species, and this is completely okay because it's specific to a project. But for our purpose, we need to only have this species. Okay. Let's see if it works without removing this. Because actually when I did this exercise, it was already different. And now has changed because this database is always evolving and there's always new data entering there. That's why this sync tag was not there. One trick that I have, because right now this came as a list, okay, so it's gonna be a bit hard to see it, as you can see. If you wanna look at it, it's like you have to go through all these different tables. Don't have a problem. See how this one there was no match because there was, of course, it's not a species called sync tag, but all of them they got a single match. I have this trick to put them all together in a data frame that is just use this function do call rbind and your list. You put it, you got a data frame with all of them. Okay. As I mentioned, you have here your identifiers for each of the species, okay, and yeah, all this data. And then I was asking you to add this data to the original data from the ETN. And to do that, we just do a merge. Using RBase, you just use the merge function. You pass the 2 data sets, and then you have to specify here the 2 columns that you want to merge, okay, because we are merging on species names. So we just add here the name of the column, scientific name, and scientific name. There are different names in each of the data frames, but it should be right. We just run it, we match all this data, and now we have a data set with all the information from ETN and also all the information from worms in this case, okay? Well, we have all these things together now. In the next exercise, I was asking you to do a special, to get the exclusive economic zone of Belgium, or the Belgian part of the North Sea. Okay, so same that I mentioned in the slides, if you use this function from marine regions and mark gas records by name, and you will get here, you can get here whatever you want, right? It's just a search thing. So it's gonna search for something, and here it will give you all the things with Belgium. We are looking for the Belgian part of the North Sea. So if we go here, we have these two. It's better if we use the exclusive economic zone, because I'm not gonna go in detail, but this is a synonym and this is actually the standard as we consider. So it's better to use this identifier, okay? 3293 for the Belgian part of the North Sea. If we put it in the next function, gazetteer record, we'll do it, and voila, we got here a simple feature object, okay? So you have here a bunch of information, and also you have the the geometry itself. If I put this here, VPNS, you can also plot it, okay, in a map directly. To do the intersection, we first have to transfer the ETN data to a simple feature object, okay? As I mentioned in the exercise, it's exactly the same. Just change that this is a single, it's not a capital letter. We run it, and that's it. We can visualize them both if we want. And voila. You will see here how all the data that we got is not only from Belgium, but it's also from all around Europe. You will see many records that are in the actually inland, which is difficult to see. And these are because of course, many of the fish, they also go to the rivers, and especially we have the eels, so that's very, very important here. But we want to get only the points that are in the Belgian part of the North Sea. How we do this? We do an intersection with ST intersection, and if you do it again, then you will see that we only got these points here, okay? This is for exercise 1, actually. I got here a bonus exercise just for you to learn. So a couple of things to mention. The same that you have your data frame with your geometry, you can also go back on time. So, and if you call st drop geometry, then what you get is just the data frame without the geometry, okay? So you can always be changing between having geometry or just having the attributes, okay? To save it, I added here a link with more information if you want to know more from the Invo code club. But the function that you write is stwrite, and then your simple feature object and the path where you want to save it, okay? Same, if you want to save it as CSV, just drop the geometry and save it as CSV, all right? Actually, this went very fast, so we have 10 minutes for questions or things if you want to discuss. Okay, I have a question from Susana as a direct message. Susana, maybe you want to unmute yourself, or should I read it? No, okay, it's a technical question about the packages and so on. No, okay. We're having a question. Sorry, where? Okay. Yeah, depending what you put here. As I mentioned here, you can call the help page of this. These are all packages in development, so there are things that may fail. So for example, there's action talks and findings, talks, detections, and so on. Well, if you go for detections, there is a big problem that you are gonna get millions of rows. So actually, you're not gonna get them. It's not. Yeah, it times out. It's not gonna load. It's impossible. I would like to fix this and try to make something that helps you to load everything. but it's at the moment it's simply impossible to download all the detections. You have to put a smaller time frames, otherwise it's impossible, because it's like this can be to the level of 10 minutes. So if you try to get 1 year, then it's gonna be really hard. So you should opt for time beans, and actually in this exercise I was using 1 week because it's better to get all it out. Yeah. Sorry. So Jonas was mentioning, the question was that that someone was trying to use this first function, the get ETN data, and in the action you have different things that you can get out of this database ETN. You can get time beans, residencies, active network, and detections. It's better to opt for time beans because this groups together all the detections. Remember that these are fish that are swimming around, and we detect whenever it passes close to a receiver, okay, an acoustic receiver. So the resolution of this data is huge. It can go to 10 minutes. Then if you ask for detections, you're gonna get a huge load of data, and it's simply not gonna work because all this data is being passed through the internet, so it's impossible to pass all that data. So it's better to opt for time beans. Then you select just 1 week or per day or per the resolution that you want, but you have more control over the data, and it's more likely to load on your R session. Okay, maybe we can look at some of the solutions here. But yeah, after this exercise, we want to have a little break, okay, for coffee, because this exercise is the heaviest. The next ones, they're going to be a bit more chill, okay? But yeah, you have any question, guys, shout, okay? The learning was still more or less the same. That's right. Yeah, she put 1 minute. It probably took more time to load, but it should be right as well. No, I don't think so. Okay, well, some questions. Well, first from Susana, but I think we answered already. This is because we were trying to get a lot of data. If you select for detention, it's better to get time beans, okay? Then someone was asking about the meaning of CRS 4326. This is a coordinated reference system, and it's I'm not sure what's the best way to show. Yeah. So this is a way of showing where are your coordinates in the wall, okay? Because you only have latitude and longitude, that actually points to nowhere. You need to have some reference to tell you exactly where those coordinates are. And this number 4326, it's a number that it's associated to a specific coordinate reference system, the wall Mercator 86 or 84, I think, and it's I will not say standard, but it's the most common one. So this is the one that forms the wall and make it like if it was just a plane and a square. I cannot go very much in detail because that's a long discussion. But yeah, just in short, it's a way of telling you where are the viewpoints in the world exactly. You can go, for example, for a projection of the Antarctica if you need to work there. If you are working very locally, you can go for a projection of Belgium specifically, or from another country. I hope it's more or less clear. Got also a question from Pablo Sanchez. So could you explain again the mean of drop geometry? Yes. So remember we started the exercises just getting some data, and when we got it was a data frame. Okay, if I run it again, you can see it. I call class ETN, simple data frame. Simple data frame thing that you get in R, your data as a table, okay? But to do some geospatial analysis here, we need to convert this into some type of geospatial data, okay? We can do that because we have here actually the longitude and latitude of these occurrences, okay? So actually the information is there. We just need to transform into a data type that are recognized as a geospatial thing. Okay. To do that, we use a package called Simple Feature SF, and the function that we use it's around here is st as SF. You put your data frame here and call it etn SF, for example. And then you have to say some things. For example, which are the columns with the longitude and latitude? So you write chords, now a vector here, C, and then longitude, latitude. And with this is enough. You can do this, and this is fine. Look how this is already another type of. Is this not? It's not. Aha, yes. ETNSF, look how this is already another type of object, and the nice thing is you can put it in a map. For example, you can do geospatial operations on it, okay? So I like to use this package map view, but you can also plot it directly or use, for example, if I try to put ETN, there is a data frame, this is not gonna work because like what you're talking about, I don't understand this. But if you pass a simple feature, it's ready to be plotted in a map directly, okay? Look, for example, here how we don't see any background, we don't see any country, as we were seeing before, and this is why we didn't apply here any coordinate reference system. So there's no way for the program to know exactly where in the world. For this is just know the X and Y axis, and then just put it on a viewer, and that's it. But if you want to tell them where is this in the world, then we have to add a coordinate reference system. The best way, just write CRS. And then the number that we were mentioning, you can look more in detail online if you want. You add this and you plot it again. Now you will see the same data, but this time it's actually located somewhere in the world because there is a coordinate reference system that tells you. So if you have coordinates and you are working with the spatial data, always have clear what is the coordinate reference system, okay? Because if you have that, you don't have any information at all. It's useless. Some more question? Yes, thanks. Okay, great. Okay. So guys, you are welcome to continue trying things if you want, but also now we want to have a 15 minutes break. Yay, for coffee. For those at home, look for your own coffee or beverage or take a nap or keep working. You are free to choose. For the ones here in the room, there is free coffee over there, and I will go in a second, so you can follow me if you want. Or you can continue here working if you want. I'll see you at 15 minutes, so a quarter to 5, to 3, so 14:45. Okay, I'll write it also in the chat. Yeah. Okay. Yeah, go for the break if you want already, guys. So Elena, instead of time links, detections, something with the species ID. Okay, I'm not sure what you mean by the species ID, Elena. Yeah, it could be that, but the AFI ID doesn't come from this data directly. But maybe paste the example on the Google Doc, and I'll have a look later. There were only 9 columns. Instead of 11. You can also mute yourself if you want to say directly, or show it in. Maybe you can share the screen.
Hi, can you hear me?
Hi, yes, I can hear you.
Okay, just yeah, instead of putting time bins as action, I don't know why I decided to write detection, and then I got a table with only 9 columns, and I was missing these species. Therefore, I couldn't go on afterwards because I was like, okay, I'm doing something wrong.
Yeah, okay, let me see. Yeah, in the example, so I did that 9 columns. You know, actually, I would like to look into that because it's when you write detections, you should be getting actually a lot of information. But yeah, of course, yeah, if you didn't get the species names, of course, you cannot go. You couldn't.
Yeah.
If you can run now the solution, you will get the exercise ready. Okay.
Yeah, I did that. Yeah. So now I realize where was the
Great.
wrong part.
In the example, you can also get the time being, so it should be all right. Yeah. Yeah. Also, if anyone had any troubles, I also did another backup in case that something breaks here, and that's yeah, maybe I will mention it later after the break. But in this folder save, I saved here the result of each exercise. So if anyone had a problem, then you can just click on it and load it, and yeah, you have that dataset to continue. Okay. We will start already, I think. How you doing, guys? How you doing? Well? Are you back from the coffee? I hope yes. It's so weird not being able to see people online. Can you see that? Okay, but you can see me.
Yeah.
Okay, that's good. Can you close the door? No one is. Okay, I hope you guys rested a bit. Let's continue with the exercise. Let's go for the second one. If you didn't finish the first one, there is a, I will show quickly. There is in the project itself. So this is same thing. There just a file explorer. If you go to save, you will see here are some files, and they have the end result of each exercise. For example, for the first one, if you click on this one, you can load the result of the exercise if you didn't run it or you didn't finish, okay? Because we will need it for the next exercises. Just wanted to mention that. Now let's continue with the second exercise. The second one, we're gonna use EurOBIS. We actually also saw some experts on EurOBIS on the room. So hopefully I don't say anything wrong. EurOBIS, we're gonna do in this exercise, and we're gonna get some extra data from EurOBIS. I will explain what it is in a second, and we're gonna just add it to the dataset that we already have, okay? But also in this way, we will see what you can get from EurOBIS and why it was so important that we did some standardization on species and regions, okay? So first thing first, EurOBIS is the European node of the Ocean Biodiversity Information System. Many of you probably will be familiar with GBIF. So this is another one, and it has a lot of data occurrences data only from the oceans. And EurOBIS is the European node. It's hosted at Vliz here in Belgium, and you can access the data from in different ways. You can access this data online through Imonet, or you can also use R, of course, an R package. We have been developing this package, EurOBIS. And in this example, I show you how you get, for example, occurrences of the Atlantic cod. As I was mentioned, this is Gadus morua as a Latin name, and this is the FID, the unique identifier of this species name in worms. So if I open it, we'll get here all the information from worms. Take some time. But the same thing, you can access this with worms, you can access this in different ways, but you can also pass this data to EurOBIS together with the unique identifier of the Belgian part of the North Sea. Remember, it was this one from marine regions. And if you pass this to this function in EurOBIS occurrences basic, there's a basic here because there are also a full and occurrences, you will see later. This only will give you around 9 columns, but you can get like up to 100 columns in information. So there's a lot of info here, okay? And all this data is standardized to a specific format. You can find more information on the EurOBIS website, okay? And if we do this, we will get all the occurrences of this species of the Atlantic cod in the Belgian part of the North Sea. And same thing, you will see here some information. See how these columns, they are adhering to a specific standard, which is the Darwin Core standard. So if you go to a website of the Darwin Core, you will see in the exercises, you will see more information about each of these column names. So this is not something that we decide to put. This is something that is standardized, and there is a specific definition for each of them, and other people in this domain are using. For example, GBIF is using also these names of the columns, all right? You can see here, you have again the same. You have a longitude and latitude, you have a species name, and of course this is standardized with worms already, and you need to do it. And it comes already with a geometry, as I was mentioning, in R it will come as a simple feature object directly. So that allows you to plot it and see how directly we didn't have to do any intersection, we didn't have to do any matching. This is already done on the EurOBIS side. So we just ask for a place and a species and we get all the information in that area. Also, of course, there are other filters you can add. You will see later, like the time that you want to get the data from, many other things. Okay, you will see in the exercises. Another thing to mention here, simply that we will use the player in this exercise. I mentioned it in the email, so many people prefer this way of working over the base R, but some people don't know it. Basic thing you need to know is that it uses pipes. These pipes are only a way of you do something and then you pass it to the next function. For example, in here, you take your data frame and you pass it to the next function, which is mutate. But you could pass it to whatever you want, okay? So in this example, I just show how to change the data type of this column, data collected. So you have a date, imagine you have a date in your data frame and it comes as text and you want it to transfer into a date type, okay? In base R, you just simply call your data frame, your column, and then you assign it to the same column in your data frame, but in the function as date. In R, in the player, you will use a pipe first, and then you will pass it to mutate that allows you to create new columns, okay? In this case, just call the same name of the column equals as date the column name, okay? There are many other things to do here. For example, you have a select. I put here the cheat sheet. I recommend you to have a look. You can select specific columns. You can filter on columns. It's very self-explanatory. And I recommend you to use this, and we will need it in this exercise, okay? And actually, I don't have much more to explain. I would like you guys to go through the exercise. This is in the same folder than before, 02 EurOBIS. And the exercise I explain it here. So in the first one, I asked you to get, for the same species that we saw in the first exercise, the same time and location, get all the information in the EurOBIS. You can see more information of this function by running the help page of the function. Then in the second part, I will ask you to play around with the package. Actually, there are functions designed to draw the area that you want to get, and you can also see the regions that you are interested in. You can click on them and see what are the identifiers for those regions. Basically, just play around with it. Mind something, these are using some services behind that we are right now in this Zoom meeting, 47 participants. So if we all do it at the same time and we all ask for all the information in EurOBIS, that's about 30 million rows, it's gonna crash, okay? So please try to do queries that are a bit more contained. Like, don't ask to give me 10 years of all the species in all the oceans of the world. No, try to keep it a bit more focused. Okay, if you don't get any data, try to just make your query a bit more relaxed. But don't try to get everything, please. Otherwise, we will have some problems. And then in the next exercise, I will ask you to wrangle a bit the data set. So you will need to fit the data set that you get from EurOBIS and the one in the previous exercise, the ETN data set. Just try to get the same columns and change the names of the columns, so we are able to just put together all the rows. Okay. You will have here more hints and more ideas. If you have any questions, drop in the chat, ask me. And yeah, that's it. I will mention these exercises until 15 minutes. I wanted to give you 15 minutes. And yeah, so we will start looking at it again all together at 15:10 test time. So in 15 minutes, okay? Hello, we are more or less done with time. So let's have a look at the solutions, all right? I have been having a look at the solutions you wrote here. They look very nice. And yeah, let's see how I solve it and what's your ideas as well, okay? So same again, I will open directly the solutions because otherwise I'm gonna take forever to write them. So I realized to having already some of the exercises from the previous, some of the data frames from the previous exercise. Once again, I call here the libraries. I get here now a unique list. In the first exercise, I need to get data from EurOBIS using the same species, time, and location. All right. So first, I will get the list of species. And actually, what I will get is the FID. I'm using the valid FID, so it's not necessary because in this case, all the FIDs are valid, so it's not a problem. But I get them here, only the FIDs I need. This case is only two, as we were mentioning at the beginning. Don't know which one it is, but the one is the eels, and the other one is Atlantic cod. All good. Then we pass to these functions. Aerobic occurrences basic. If you have tried EurOBIS occurrences, you will see here a few stuff. So you have here other options, and in the bonus exercise we will use some of them. Okay. So we're gonna use basic to not overload the server. And we're gonna get for this region, that is the Belgian part of the North Sea that we used before, if you remember, then the list of VIDs and the same dates, okay? We do this and we get basic data. So it's just a few occurrences, but we can increase this by adding other regions. That's why in the next exercise I ask you to just try things out, okay? Some information you have here, you have your and the dataset ID, so you can go to this URL and you get more information about each specific dataset, how they collect the data, why they did it in a certain way, etc., etc. Who paid for it, all this type of information. You have here your latitude, longitude, species name, the ID from IFIA. It is important that in this case it doesn't come as a number, it comes as an URL, so later we will have to get rid of this to have the same type of field as in the ETN dataset, okay? And the geometry is a simple feature object, all right? So the next exercise, I'm not gonna go much in detail, but I ask you here to just select your region of interest, this locally, and it allows you to draw a polygon of the area of interest. So yeah, if you just select all Europe, you can do it, but then of course this is gonna be a big polygon, it's gonna take some time to load. So I don't recommend it. I'm not gonna go much in detail, but you have here all these functions that you can try. Okay, I saw there was some problem with this one. If you call this one directly, just this without the ecoregions or something, then yeah, it's likely to give you some problems. Just saying here, you wrote here EurOBIS map, then you will have here a lot of options. If you go for regions, if you go to the help page of each of these ones, you will see more information. These all come from marine regions. This, for example, are the International Hydrographic Office areas. So this authority, it tells you where starts the Atlantic Ocean, where it finishes, where is the North Sea, where is the Indian Ocean. It tells you all these things. It's an authority. We just follow the recommendations, and we can plot it here in a map. Okay, if you do this, it will open a map that allows you to just click on them, and you will see the identifiers of each of the regions. Okay, it may fail sometimes because it relies on internet things, so it depends. If servers are not available, it might not work, but in this case it's working. You can see here, you can see some information, including the marine regions ID. So if you use this in another function of marine regions, like we were mentioning before, MR gas record, and you add this here, then you will get this polygon, okay, this area from marine regions. You wanna do it? Just to mention, and here you can try different things. For example, you can also query on functional groups and many different things. As you may see, I would like to show you quickly occurrences, basic. You have different things here you can query on. For example, we have not only functional groups like birds, mammals, benthic, whatever is interesting for you. We also have, for example, the UC and red list, so you can select like if they are at least concerned, if they are endangered, and other things like the CITES annex, ITAS directive, etc., etc. All this information comes from worms, and this is very powerful in this package because it allows you to get not only taxonomic but also functional groups and all this type of information. And the next exercise, what I was asking you is to, well, you have to clean up the data and make them look the same both in the ETN and in the EurOBIS data sets that you got in the first exercise and this one. This is actually not the scope of this workshop to show you how to clean up data, but it was necessary for this. So I hope you already had some knowledge. If not, while you're using here some functions from the player package, as I mentioned, you pass your data set to the next function through pipes. And here I select a few columns that I was mentioning before. So just selecting a time, a species name, together with the FAD, of course, and then a longitude and latitude to know where they are in the world. I had to do this trick to get rid of, as I was mentioning, of the whole URL of worms. So I get only the number, the FAD, okay, the identifier of the species. So if I run this, if I run this, then I do this again. We got only these columns, okay. Then in the ETN, there's a trick that is you can use select to select the columns and then mutate to change things on those columns. But there is also a nice function called transmute that it allows you to transform things already on these columns, and already you just select those columns. So I was doing this, okay. So what the only thing I'm doing is in the ETN, you remember they have a few columns, and I just want to have the time, longitude, latitude, and scientific name. But of course, it's called like scientific underscore name. Longitude doesn't come with decimal longitude, doesn't come with decimal latitude. So what I'm doing is just changing these names here. So I'm changing to date collected, to decimal longitude, decimal latitude. These are all, as I mentioned, standards of Darwin Core. You can find more information here about each of them, right? Then because all this data, it comes from detections, and we want them to happen only by date, there's gonna be more than one row per day. So we don't need all that extra information. We're just gonna keep a few of them. So once we got rid of all the other columns, this will be duplicates in the dataset. So if we use this function, unique data frame, and then we will get only those. This works better with simple feature objects. You could use the dplyr distinct, but it works worse here. That's why I'm using this one, okay? And at the end, what we do is bind rows, is we just take all the rows here and we add it to the data frame here. So if I run this, you can see, well, this is actually what I wanted from the exercise. So what we got, it's a data frame with data from ETN and also some data from EurOBIS, okay? They all have the same type of information. They are all here together, okay? Bonus exercise, this is a more tricky one, okay? This is a bit more difficult, but this is just for you to be aware of all the information in EurOBIS, in the EurOBIS package. So there is not only, of course, they don't only just the species names, the location, and the time. There is all the information like counts. Sometimes there is information on the life stage, information about how it was sampled, about many things. And all of this comes with the full and parameters. So if I repeat this with full and parameters, you will see that I got more rows than before, and many more columns, okay, up to 100. And here you will see some of the columns are called parameter, okay. These parameters are, so these are how we receive the data from the original provider. For example, density, count, but yeah, sometimes they can use, for example, a capital letter. In other ones, they don't use a capital letter. Also, how do you define density? How do you define abundance? Okay, so we standardize this to another standard. It's called the Biodiversity terms. The Biodiversity is the British Oceanographic Data Center. They have a list of already defined terms that are interesting in research, especially in oceanography, right? Example, instead of just density, we have abundance of biological entities specified elsewhere. per unit area of the bed. So it's very specific of exactly what we want. It's very specific with the units. So what we're gonna try to do is we are gonna pivot this table and we're gonna convert these terms here into columns, okay? We actually will use, I'm not sure right now. I think we will use, what we will see here. So first in this I just showed the different parameters available for this data frame. Okay, for example, we have bed abundance, count, gender, the instrument used in the sampling. It depends on the data set. Different data sets, they collect the different data. So not always you want to have find a lot of NAs because not always you got the same information for all the data sets. But we include them all in EurOBIS. So in here, what I'm gonna do is I'm gonna get rid of the geometry because it's gonna give us a lot of problems. Then I'm gonna select a few columns. I'm gonna go the name, the date, longitude, latitude, then these two columns, parameter and parameter value. Parameter is, as the name says, the parameter, so it can be count, it can be abundance, it can be gender, but this is already standardized. And then also the value for this parameter. Okay, I get only this, and then using this function from TidyR, I think it's called pivot wider. Then you pass these arguments, names from parameter. This means that the names in the parameter column, they're going to be transformed into more columns. And values from is what are those values going to be? So the values are going to come from parameter value. If I do this, then you see I have the scientific name, the date collected, decimal latitude, decimal longitude, and now here I have a column for each of the different parameters in there. For example, we get here the bed abundance. You can find more information there. The count, gender, instrument. Okay, you will see there is a lot of NAs. There are nulls. Why is this? As I was mentioning, this data, the data sets, the people that provide the data set can give different information depending on their research idea. And we collect them all together, but of course we don't have all the information available for all the data sets. But this is the best way we found until now to put together all these different data types and to share everything, okay? These solutions are online, so you are very welcome to use them for your own purpose if you want. If you have questions, you can also write us. Actually, that's it for this exercise. If you have any questions, we can have a look at them now. Feel free to drop in the chat, to unmute yourself. If you are here, just say. Otherwise, we'll wait 1 minute more, and if not, we will go to the next exercise 3, okay? After the exercise 3, there will be another break, don't worry. The exercise 3 is not gonna be that long. And, yep. Okay, I'll go for the next exercise 3 then. I hope you guys are fine with that. If you have any questions, you can come back later. Also, this workshop is supposed to finish at 4:30, but I will stay half an hour longer if you have more questions. So there's things you would like to check out. Okay. So in the exercise 3, I hope you are seeing my screen now. Okay. So what we're gonna do is we are gonna get now environmental data from other databases. Okay. Keep in mind that the same that I show you here. Okay, it's not showing exactly my screen now. Hold on a second. This is a zoom thing. Okay. Now you should be seeing the slides now. So in this exercise, we're gonna get environmental data from 2 other databases. Okay. Keep in mind that you can use this for all the databases that are around. These are the principles, and then you can reuse them for their own purpose. And yeah, so we're gonna get environmental data from BioOracle, and also okay, one second. Yeah, we got our host saying that he was leaving. So we're gonna get environment temperature data, for example. We're gonna get seabed data, for example, from two different databases that are BioOracle, that is funded by LifeWatch, and also from EMONET seabed habitats. We'll explain a bit more. So the first thing I want to mention is that the geospatial data can come in two different forms. For example, up until now, with the data sets that we have been seeing transforming into the simple feature thing, this is vector data. These are data that comes with coordinates, with exact coordinates in the world. Where do we find this? Like in this point, in this point, in this point. Using those points, for example, you not also have points, but you can also draw a polygon. For example, in this picture here, you can see this is one node, this is another node, this is another node, another. And this is how you draw a polygon of an area, okay? This is very nice and also allows you to have not only the geometry of the area, but also attributes. And these attributes in R is what we have been seeing is the data frame that is in that simple feature object. For example, the species name, time, etc., etc. So we're gonna get some data that is in vector format, but also we're gonna get some data that is in raster format. What is raster? Raster is basically an image, the same image that you have Instagram, the same pictures you put there. Those could also be theoretically called raster. The important thing in these raster layers is they come with information also associated to each pixel of that image. So for example, a very common use is to have the temperature. So you have a picture in which each pixel has a value for a temperature. So if you paint, then you add colors to that picture, and depending on that number, then you get a map telling you where is hotter, where is colder. Okay, but the principal difference is that one with vector layer you have an actual geometry with points which tells you the coordinates exactly, and raster data is an image that is referentiated to the world, but it only has information for one variable, okay? Normally. We can discuss that, but normally it comes only from one variable. So in this exercise, we want to get data both in vector format and raster format, okay? And then in the next one, we'll learn how to extract those data, the data exactly that we want out of those layers, okay? So first, we will get some data from EMONET. European is let's see if I see it wrong, the European Marine and Observation Data Network. Okay, it's an European project. It has 7 different lots with different data, both for biology, for physics, chemistry, human activities, civil habitats, etc., etc. We have been developing in the EMONET biology lot. We have been developing an R package, which is the EMONET WFS, that allows you to query all the vector data from EMONET. Okay, when you see this random thing WFS, when you see the F, it means that it has vector data, which this actually means web feature services. I'm not gonna go into detail because it's a technical informatician thing, but when you see WFS, it means that it's vector data. Okay, we will later in the future we have also a package for raster data, but until now we have this one. If you load this package, you have a function to see all the services that are there at the moment. So you will see the service name and also the URL to the services specifically. You don't have to worry that much about it, but as you can see, you have different lots. We are going to be looking at the seabed lots for this exercise. Okay, so to call all the layers and see what is in this service, in the seabed habitat service, you have to first create a client. You don't need to know much details. It's like an access point. So you first use this function, EMONET init WFS client. You add this text that you can find again here when you call this function. And then you create a client. You will see that it's a different class. This is not something that it's seen very often, but you don't need to query that much about it. So you have this client created, and now you can pass this client to other functions to get other types of data. For example, if you pass it to EMONET get WFS info, you can see all the layers that are in that service, okay? And you get some information from there. For example, if you do this, you will see the service URL and then some information like the title. You will see the abstract to get more information about it, and the format that it comes, again, SF. And what it's important here to keep getting more data is this layer name here. So once you read the titles, you read the abstract, you select one layer that you're interested in. For example, in this exercise, I think I was getting Let me see. Let me see what it was. 1110. 1110. It is. Yeah. This one here, which is actually sandbanks. Okay, so I'm gonna get a layer that tells you where are the sandbanks in Europe. If you take now this layer name, whatever it comes, it doesn't matter the name, it's just an identifier, and you have the explanations here. You pass it here to another function, which is immonet get layer. Then you have to pass the client, you pass the layer code. Again, you should say the coordinate reference system that you want to use. In this package, if the layer is not natively in that coordinate reference system, it will be reprojected on the fly. So you don't have to worry that much about it. Just pass the reference system you want, and that's it. You pass it here, and then you get your layer, and you get it as I mentioned before, a simple feature object, and it comes with a bunch of information here. Most important for this, in this case, it comes with the geometry that you can plot. Okay. Well, you may have to use this later. This is a little trick because it comes with a specific type of geometry that's a bit complex, but if you run these two things, it will transfer into polygons, so you will not have any problems, okay? So once again, I haven't tried with the presentation, but this should look better in Urare Studio. And you have here all the polygons with all the information of this area. For example, in Belgium, basically all the Belgian part of the North Sea will be sandbanks, or they are considered sandbanks by a specific authority. Now, this is about the vector data, the sandbanks, but we will also get more information in raster format for Bioracle. Bioracle, it's a project that creates data layers that are using ecological modeling. So it gives you different environmental variables like temperature, salinity, chlorophyll, pH, I don't remember right now, current velocity, a lot of different environmental variables, okay? And it gives you for present conditions, but also there are future forecasts for different climate change scenarios. So this allows you to do species distribution models, both for present conditions, but then also extrapolate for future conditions and the climate change scenarios. And in R, at the moment, you access this data with this package SDM predictors, okay? If you want to get more information, it's also here in the website. What we want to do here is we are going to load, well, we need to load first SDM predictors and also this other package, raster, okay? That this allows you to work with raster data. To see the layers available in Bioracle, you run this thing here. You have to use the last version, is 2.2. And what is important again is use the layer codes here. So you select the layer that you're interested in, and you copy it and you pass it to the next exercise, to the next function. Okay. These are the layers for the present. For the layers of the future is the same thing, but layers future. So same again. You collect here all the layers you are interested in. This bunch of stuff here is explained, but these are the different scenarios. So RCP 26, you have 26, 45. Etc. etc. Two more of them, okay? And also for what year, 2050 or 2100. Once you have your layers ready and you know which codes are you going to use, you pass it to the next functions with these load layers. You can pass here actually not only one, but you can pass 3 or 4 or 10 or whatever you want of them. If you add here a path, it means that it will be saved in your computer, so that's very nice. You don't have to download them again and again and again every time. And once you have this, you will have your data as a raster layer, okay? This means that every pixel has information on it that we will extract later, okay? As I mentioned, you can, this is a raster format, so you can just plot it directly in R. I mean, this is how it will look like, for example. Voila, after this, I will just let you to go to the exercises. It's just 2 of them, and it's what I was mentioning here, but they're asking different things. You should not have much problems, okay? It's as I mentioned before, in source exercise 03 environmental. Yeah, go for it. Yeah, didn't mention the time, so let me see. We're a bit behind schedule. So I would like you to give you 15 minutes for this. That's come back at 3:50 more or less, okay? All right, guys, it's 10 to 4. Okay, let's have a look at the solutions. I hope you got there more or less. This one was a bit shorter, but it's not. It's okay. You have always a solution there. Really? Okay. Yeah, that's the problem. Depending. So one of the colleagues here was trying to load one of the layers, and yeah, it has been loading for 5 minutes. That's very possible, depending on the detail of the layer. That's why these exercises are designed to be a bit contained, but this can be always be a challenge to load a lot of data. So once again, I will open the solutions. Okay, you can find them as you know exercise solutions, but if you come up with something different, that's perfectly okay. I will load these libraries, and first we'll get data from Emonet, using the Emonet WFS package. Okay, so we have to start a client, as I mentioned, explore what layers are available, and then I ask here to get either reefs or sandbags data. We'll get the reefs data. Okay, so actually this is you first need to see what the services are available. I know already that because we saw also before that this is the name for the service we are looking for. So they have in seabed habitats, they have 2 different services. I don't know exactly why. You can always send an email to them. We are not seabed habitats, so we don't know. But the layer that we want is in this service specifically. Okay, so we first create a client with this function, Emonet init WFS client. It may take some time, but it normally loads. Okay, no problem. Then we want to see what are the layers in this service. So we use Emonet get WFS info with the client. Now, if we go to see this, it's exactly the same that I showed in the slides. So as you see here, all the services that are in this, all the layers that are in these services. Okay. So since we are looking for reefs data, you can look at the abstract, just need to read them, look at the titles, and here already. Okay, I pushed something that I should not have pushed, but anyways, the layers are here, and we are looking is for the reefs. So the reefs, voila, it's here. But you can see that it's also Posidonia, beds, mud flats, estuaries, coastal lagoons. All of these, they have a purpose. You can read more about them here. So you select exactly what you want. In this case, we want reefs. So if we go for the layer name here, we copy, we go back to exercise, and then we leave it somewhere here. And then in the next one, we need to get actually the layer. To get the layer, we use Emonet get layers. You pass the client in the WFS argument, you pass the layer, and then there are here some different stuff. The CRS, the coordinate reference system, as I mentioned, is important. We want them to have the same coordinate reference system as the data from EurOBIS and ETN. Okay, so we are gonna ask for this one. You will see actually there are some messages because the coordinate reference system of these layers is not this one, it's not World Mercator, it's another one, but the package itself, it's able to reproject them on the fly. Okay, and also we will use this option here, reduce layers. Okay, here there is some things I need to do because it comes with a different type of geometry, but it's not a problem. It just want to convert them into polygons. Okay, so if I run this, we will get the layer. It'll take some time, hopefully it loads. Okay. See, for example, it tells you here that the coordinate reference system is being transformed because it comes from another one. Okay. Of course, once you have this, we will load both in a map the data frame before and this layer. We can put them all together and just see them. And voila, we have them both ready to be plotted somewhere and ready to be extracted. But that's what we will do in the next exercise. And we can spoil it already. So we will get these points, we will get this layer, and we will extract values from the layer using these points. Okay. Now we have the vector layer ready. That's fine. Now we go for the raster data from BioOracle. Okay. I was asking here to get the mean sea surface temperature from BioOracle, from present conditions and future conditions using the estimate predictors R package. Okay, so as I was mentioning here, you can see all the layers available using this function, list layers. So you select the project you want to see by Oracle, and the latest version is 2.2. So you can do this, you can do this other one. and then you can see all the layers there, all right? Since we are looking for temperature, one idea, for example, is just go to the name and write temperature. See, voila, 0 the temperature. So since you are looking for surface, we can write surface temperature. And we are looking for the mean temperature. So voila, here, see surface temperature mean. And here is the layer code. So we copy it, we keep it somewhere. We do the same for the future layers, okay? In this one, since we have already the current and the layer for the present, we can also paste it here and we will get all the layers that are for the future in here. I was asking in this exercise for a couple of the, for the future scenario for climate change, RCP 85. You can find more information about this on Wikipedia and many other places. For both for 2050 and 2100. So I get the codes for those. I wrote them here already. I get here all the codes. And as someone was mentioning before in the chat, yes, you can load many layers together. You just pass them to this function, load layers, all the codes. And in here there are different options you can write. For example, data dir is where you're gonna save the data. I'm gonna do it already because it takes some time to do load. Raster stack, it tells you it is a type of raster object in R, so you can ask for that one. It takes some time, it's to load the layers, but it should be done very soon. Okay, now we got all the layers ready. They are all already in this object, but they also have been downloaded to this path, which is in the project. It's in data, under data, okay? So you can see here different things that have been downloaded, and these three are here, okay? So you can also not use it only for R, but you can also use them as a way to download the data and you read them later in QGIS if you want, or whatever other place, okay? Voila, this is the exercise. Then the rest, it was just to show you that, yeah, we got the layers, eh? So you can also upload them directly. They are in raster format, and in the next exercise, we will use them.
Someone asked in the chat if you can also plot the EurOBIS layers in plots or base cells.
I know you can do it. I don't have much experience, I have to admit. So that's why I'm sticking to. So yeah, the question, I don't know if I will repeat it. Could you please explain other plotting options for raster layers loaded from BioOracle, example using base or ggplot2? So in my case, I'm not very familiar with those. I know that it's completely possible to just plot them with ggplot, but I'm not very experienced with that. So that's why I was using this system, because it shows the layers quickly. I know that if, for example, you want to show one of them, you just directly like plot layers. It's probably not going to work for different reasons. First, because we are calling 3 layers together, so we have to plot only one of them. Layers. So this is not ggplot, this is base R. So you have to call only one of them. But still, it's probably not going to work because it is using the base plot. And it works different than the plot from raster. So the raster package has its own plot. So you have to first call the package, 2 points to points, plot, and now you can plot the layer that you want to see. Layers, and the first one, for example. In this case, you can plot it with base R. With ggplot, I don't know right now. We can look into it. If you're more interested, send me an email. We'll look it up. Which is BioOracle? I'm not sure by heart. I think it's 0.15 degrees, something like that. I'm not sure. We can look it up in the website. There is also 2 papers associated to this. Let me see if I can show you. Also, I can let you know already that we are working on a 3rd version of BioOracle that will be ready soon this year, and it will have a much higher resolution. And actually, you will not be able to download a whole layer in one because it's too heavy, and you will have to download only certain parts of it. And here on the website, you can find more information. Example, voila. So the resolution is 5 arcmin. And but also you can look here and you can find that there are some publications. I'm not sure exactly where they are, but there is frequently asked questions. You will find somewhere here. Yeah, in the site you will find there are 2 publications that explain better the data set. Okay. Voila, so this was the exercise 3. Let's have a little break, okay? For actually we got here 15 minutes, but let's try to keep it on 10 minutes because we are behind schedule. Actually, we're gonna finish a bit later than the hour that we're supposed to, but it's okay. Also, we have this questionnaire, there's a survey with some questions. Please, if you can take some time to answer it, either now or later today, it will be very helpful to us to see, like, if you like this workshop, if you would like other type of content, if it was too fast, if there were different things that we can improve. Okay, we are also learning how to do this type of workshops, and so in the future we can try to make it better, of course. Now only just 10 minutes. Let's come back at at 16:10, and yeah, you have time to have a quick coffee. That's great. And yeah, if you have questions, you can always pass them in chat. We have a look later. All right. Okay, hello everyone again. You had a nice break. It was short, but yeah. So this meeting is supposed to finish at 4:30. So in 20 minutes, we wanna have to take a bit more of time. That's okay because we have this room until 5:30, I think. But we try to keep it short anyways. But if you wanna have more questions later or look at things, I'm happy to stay here a bit longer. All right, both online and in the room. So let's go directly to the last exercise. It should not be very long. So in the last exercise, we're gonna do it's combine things together. So we're gonna combine. Let's think about it again. So in the first exercise, the first exercise we got data from LifeWatch. If you remember from the ETN, so fish data. We made a match with worms and with marine regions. Now in the last exercise, in the second exercise, we got data from EurOBIS. We put them both together, LifeWatch and EurOBIS data. And in the last exercise, exercise 3, we got data from InmoNet and BioOracle. So we got seabed habitats and temperature data. And now what we want to do is put them all together. So take the data points from both LifeWatch and EurOBIS and match it with data from seabed habitats and BioOracle. So we will know what was the temperature in this point, what will be the temperature in the future in these points, and also we will know if they are in a reef or not. Okay. So in this last exercise, we want to do that. Technically, what this is called is a spatial joint. So imagine these are all the data points from the data frame that we were working on. Okay, then we put here a layer. If you know already, this is the Belgian part of the North Sea, but it doesn't matter. It's just an example. And what is going to happen at these points, we'll get the attributes or the values of those layers. Okay, so in this image I just made it as if it's paint on blue, so it's inheriting all the attributes from this layer. That's what I want to do, and this is the concept of a spatial join. So we want to join the attributes by location. Okay, this is different either if you're working with vector data or with raster data. With vector data, we go again to the SF package, we do stjoin, your data frame, your layer, bang, you got the things joined. Okay, if we are actually, but instead if we are working with raster data, we have to use one of the functions of the raster package, which is called extract, and then we passed the raster layer, and then we pass the points. The points we have first to convert them to another data type called spatial. Doesn't matter, just call this function sf as a spatial, and then you pass it to the extract, and then you raster layer in those points. Okay. Well, that's the exercise. So you have to do these two things. And actually at the end, you can have a look at these data sets already finished and get more ideas of what you can do with it. Think about it, I don't know, try things out. Yeah, we will be done. Eh? So yeah, actually I will give you 50 minutes to do it. Then we can go through the solutions, but it should not take that long. But we can use that time also to discuss other ideas. Yeah. Also, don't forget to fill, please, the questionnaire. I will say it again before the end of the meeting. We'll send an email also because it's really nice for us to get some feedback on this meeting. Okay. Okay, it's 4:30. So let's have a look at the solutions, okay? And then if you want to stay longer and have more questions, try things out, I'll be here a bit longer, okay? But for those that need to leave, then we will finish this already, and it's okay. I will share quickly my screen again. Okay. So once again, I'm opening the solution. In this case, it's gonna be quite short. So we need these two libraries. They're already loaded, but just in case. So in the first exercise, I asked you to make a spatial join between the seabed data that we got in the exercise 3 and the dataset that we compile between exercise 1 and 2. All right. So as I mentioned before, this is a spatial join, and in R you just use st join to do that between the two data frames. Okay. One thing I'm gonna do first is just to select the habitat description column in this dataset, because if we look at it, there are many different columns that we don't really need. And it's all interesting to have the habitat description in this case. If you were interested in something else, you are free to keep them. And in this case, I'm just interested to know if they are in a reef or not. Okay. So I will just keep that column. I'm using the plier for that. I select the column, and then I do stjoin, data frame, seabed. So in this one first, we can have a look first if you want, or even better, view df. You can see here we have this this information, but when we do the join, if we do the view again, it's one extra column that is the habitat description, and we can see which one are in a reef and which ones are not in a reef. Okay. If you are interested in something else, depending on your research question, I select another habitats or select another layers. These are just the principles. You can apply them for whatever you prefer on your research. In the next one, we need to extract temperature data from BioOracle. We got them before, remember, in this variable in layers. Okay, we have 3 of them together. You can access it this way. Just write layers $sign. The 3 of them are here. Okay, so for that we are gonna use the extract function from the raster package, and then we pass the layer itself, and we need to pass as well the data frame, but it needs to be converted to another type of spatial points. Okay, it's a requirement of this package. So we convert them, and then we extract the values there. And directly what I'm gonna do is assign them to a new column in this data frame. Okay, this column data frame df $sign temperature doesn't exist at the moment, but once I call it and then I assign a value to it, it will be created. Okay, so if I call this alone, it will give me just a list of a lot of numbers, and these are the temperatures on the sea surface for present conditions. Okay, on those points. If I call it here again, and we do view df, you can see that we got a new column with all these temperatures here. They're associated to a species, to a location, to an habitat description, and to a time in particular. Okay, we can do this also to the future condition layers, and voila, we got our data frame with our date, the species of interest, the location. They are interconnected via FID with worms. We know that they are only in the Belgian part of the North Sea. They are connected with marine regions that way. And also we got extra information about temperature, both for present and future conditions. Because you can see here that the temperature for the future is gonna be higher. This is anyways a prediction. And that's it. You can do whatever you want with this, or add new variables on BioOracle, or from other services. Copernicus has other data. I'm sure NOAA has other data, and these are the principles that you can just use them for whatever you want. And actually that's it. This is the end of the workshop, and I hope you guys enjoy it. And if you have more questions, we can talk now, or always you're very welcome to drop me a line on the email, or also I can show quickly in the repository itself. If you are used to GitHub, you are very welcome also to just go to the issues and open an issue here, mentioning what didn't work or something that you would like to clarify. The slides are here online. All the materials are online. We will upload this the recording of this session as well, and we will put the link somewhere here. Yeah, and that's it. And please remember to please fill this this form. We got already some replies. They're very useful. I saw, for example, there was not much time. That's true. It's a lot of content for 3 hours. We'll try to get more time for the next one. But anyways, all the materials are online. You can try on your own. Okay, later. Got the comment from Pablo. Need time to digest all the info. Yes, I know it can be hard. I'm a bit tunnel vision right now because I'm doing this all the time, so it doesn't seem for me that much, but I understand. Do you know a similar database to ImonNet for marine habitats, but at a global scale? No, I'm not sure. I could say you can have bathymetry from GEPCO, but I'm not sure. Maybe you can try in NOAA or in maybe in Siemens, in Copernicus. I'm not sure. If someone in the chat knows, feel free to unmute. Okay, I'll send the link to the questionnaire via the chat. No problem. So we'll stay a bit longer in the meeting if you have more questions or whatever, 10 minutes more. But if not, we will close it. We are free to leave already if you want, guys. It's okay. And thanks all for coming. We got about 50 people and the online meeting, about 10 people here live. We got 100 registrations, so 50 of them dropped. But I was expecting that. It's not a problem. But it's great to see so many people here engaged on these things. And if you find any problem with any of the packages, we are developing most of them. So feel free to message me. We will open issues on GitHub. We can actually change them. Yeah. We got a question here from Elena. How can we normally access the packages that are under development? Good question. So normally on GitHub, if you see my screen, we're here. Let me show, for example, I know that the path already. Okay, so that's the first problem. Maybe if you don't know where is the package is a problem. But most of them they are in LifeWatch. For example, we are developing this one at the moment. It's a second version of marine regions. So you go to the path, and normally it's explained here even how you can do it. Normally in R, you just use this other package, DevTools. You call DevTools, install GitHub, and you just need to give well, actually you can see here, it's this path here, right? So you just copy this, run it in R, and it's installed. So if I try even out of the blue here, yeah, I'm not sure you can see my studio session. but just copy this. No, you cannot see your line, but it's
Where
okay. Yeah, I know, but it's okay. If you just pass this to the RStudio, you can install it. You can also, if you have a GitHub account, you can watch it. So every time there's a change, you will be notified on your email. Yeah, so this is the way. No worries. Okay, for those that are still there, thanks a lot for coming. We'll close already. If you have any questions, as I mentioned, you have my email. You can guide me, you can guide any of the team. You can open GitHub issues. Yeah, I hope you enjoy and it was useful, the workshop. Thanks for coming, guys.